Hello, Ms. Gina. Today our group will present about edaphic factors and plant influence denitrification in soils from a long-term arable experiments. So here's our group mates. So what is denitrification? Denitrification is the process of microbial reducing nitrate and nitrite in the form of nitrogen gases such as nitrous oxide and nitrogen. Moreover, the emission of greenhouse gas due to the bacterial denitrification contributes to global warming. Nitrate can act as an alternative electron acceptor and is reduced to nitrous oxide by anaerobic bacteria. This occurs with different types of fertilizers. In a survey of genome sequence prokaryotes, 7% contain denitrification genes which are near K, near S, and no Z. However, not all prokaryotes with a nitrite reductase gene have no Z, and also the other gene is not active. Consequently, for these organisms, the final denitrification step is the release of N2O. Also, there are also bacteria that contains no Z without near K or near S, and assumed to reduce N2O emitting N2. Based on the survey for NO2 fluxes, it was shown that there are few investigations towards the nitrogen emission and the role of the crop plant. Hence, the aims are to measure the production of nitrous oxide and nitrogen in contrasting long-term treatment soils to anticipate the alteration of microbial communities that affect the nitrification genes and the effect of growing plants and applying KNO3 fertilizers on grass production.
In these experiments, different fertilizer and N rates have been applied consistently to winter wheat. We chose plots that receive farmyard manure, no N fertilizer, and 288 kg of N, together with woodland soil. The total of 24 pots planted with pre-germinated wheat seeds and the 24 unplanted pots, each with different treatment, with or without wheat and N as fertilizer. So for the method, soil, pot, and plants were prepared. Then, the second one, when plants reach stem elongation, the soil were adjusted to create the anaerobic condition conducive to denitrification, creating closed chambers. Then continues by gas sampling and the measurement of nitrogen and nitrogen oxide. After that, the DNA and RNA extraction and amplicon sequencing were conducted to find the bacterial and archaeal diversity of the samples. Then, quantification of bacteria and denitrification gene abundance conducted to find the relative mean abundance of prokaryotic phyla or subphyla. Soil properties measure moisture and estimate the final WFPS. Last one, statistical analysis conduct to compare values obtained from soil and gas analysis.
The result shows that the origin soil is the most significant influence on soil properties, gene abundance, and gas production, especially in woodland soil, while the presence of plants with or without has various influence, and the presence of fertilizer with or without again has the least influence. Correlations are observed in denitrification genes with bacterial abundance and ammonium. Nitrous oxide has no relation with denitrification genes, but it correlates with the nitrogen isotype, water-filled pore space, and strongly relate with nitrate.
So now there are going to be three major points that we discussed in this paper. The first one is the role of microbial diversity and abundance. This is affected by two things, which is the soil treatment and the 16S rRNA gene abundance. Different soil treatment will result in different conditions that favor different phyla over the other, and this includes pH as well. For the 16S rRNA gene abundance, microbial biomass related to the NRK and NUSZI, which is unaffected by nitrogen addition, does not affect nitrogen gas and nitrous oxide production or denitrification genes, which is shown from the gene proportions. When nitrous oxide reductase increase, nitrate reductase doesn't. Therefore, less nitrous oxide will be formed because more is converted to nitrogen gas. As for the inaccuracy, 16S rRNA gene abundance was not found to be significantly lower in bare soil compared to planted pots containing reservoir soil. This is because the qPCR analysis lacks the data. So the second part is the importance of plants in gas production. The first one is because gene abundance does not affect gas production, which does not affect denitrification. Because of this, low nitrous oxide to nitrogen ratio means that less denitrification occurs. The NH4 plus N- ions formed is a product of soil organic matter mineralization, or SOM mineralization, and this can be stimulated by the rate-short exudates. Lastly, more nitrous oxide and nitrogen formed when plants are present because nitrogen gas increases more nitrous oxide is denitrified, might be caused because of the closed system, which will result in prolonged contact with microbes. And the experiment uses mature plants because the denitrification is more visible. Lastly, it's the soil factors that drive denitrification. There are a few limitations of this experiment. Because of the closed system, it has a maximum concentration, and the presence of air, if it's not in closed system, is hard to control. Other than that, acetylene inhibition that are used to prevent nitrous oxide to nitrogen gas is also disadvantageous, and the need to sample manually is also disadvantageous. To improve, you can use the robots and other factors in future experiments like moisture levels, sequence amplicons to reveal different groups to estimate gene abundance, and monitor the gas emissions. Secondly, wheat takes more than the available nitrogen for denitrification, more than the maximum amount of conversion, which is 4%, which might be the cost of enhanced disappear effect. Thirdly, in all of these pots, the nitrous NO3 is evenly distributed. And lastly, the factor that drives denitrification is nitrate availability. That shows that plants promote denitrification and genes do not really play a role.
In conclusion, the study demonstrated that the concentration of nitrous oxide was higher when there was addition of fertilizer, and otherwise, if there was presence of plants, the concentration was lower. The dinitrogen concentration was increased over time with the plants. Prior long-term soil management influenced the soil microbiome and also the denitrification genes. In anaerobic condition, which is the experiment condition, the production of nitrous oxide was driven by the nitrate availability, and also the nitrogen generation increased in the presence of plants. Here's the reference that we use, and thank you for listening. Have a good day.